In this video, we're going to be learning about the ventricles and the structures associated with cerebrospinal fluid. Let's take a look at our ventricles first. This model is showing the ventricles of the brain. Remember that the ventricles are empty spaces that circulate cerebrospinal fluid. So the gray you see here is actually showing you empty space. The top ventricles here and here are kind of horseshoe shaped. They're known as the lateral ventricles. They're circulating cerebrospinal fluid through our cerebrum. The lateral ventricles dump down into the third ventricle, which has four sides and a hole through it. That hole is going to be important here in a minute, so don't forget about it. The third ventricle sits between the two halves of the diencephalon. Draining the third ventricle is this passageway here, known as the Cerebral Aqueduct. The Cerebral Aqueduct carries CSF from our third ventricle down to the fourth ventricle. The fourth ventricle is this structure down at the very bottom. Now you'll notice when I turn the ventricles over so we look at their undersides, we see all of this pink here. That's meant to represent the choroid plexus. Choroid plexus is essentially ependymal cells sitting on top of blood capillaries, and that's how we produce our CSF. So the choroid plexus makes the cerebrospinal fluid. Now if you're looking at this model and you grab a brain, you're probably wondering, how does this go inside of this? And in order to help you visualize it, I've taken this model and broken off half of the ventricles. Now notice that third ventricle has the hole in it. If I look at my brain, I can see the thalamus has this kind of brownish gold dot. That's called the inner thalamic adhesion. That's the connection between the two halves of the thalamus that runs through the third ventricle. So that dot matches up with our little hole, and we can see that the ventricles would be sitting just like this inside of the brain. So our third ventricle would be the depressed area in between our two thalamuses. We see here, that's our cerebral aqueduct, and this space here between our cerebellum and brainstem is the fourth ventricle. Now a couple other structures that we need to mention are the arachnoid villi and the dural venous sinuses. So we're going to take a look at this model here. Now running around the surface of the brain, we see these big blue spaces. These are the dural venous sinuses. The dural venous sinuses are actually venous blood vessels that are formed between the two layers of the dura mater. We can also see, if we zoom in very closely on the model, these little green balls. Those are meant to represent arachnoid villi. Now your arachnoid villi in you and me and anyone else who's alive is not green. Why they decided to paint these green, I don't know. But the purpose of the arachnoid villi is they allow CSF from our subarachnoid space to flow back into the venous blood in the dural venous sinuses. This allows us to return CSF into the venous blood flow.